just temporary. 30 meters, I see a hill. Look at, hark, hark.
What phase are we in with the moon? Or like the tides right now?
We just passed full moon. I think we had full moon just a few days ago, didn't we?
Seemed like it, yeah. So for those who don't know, we have pretty minimal tidal changes here in Hawaii by comparison with other areas. You know, it might be a foot, might be, if that, can be more than that. What's our max tide like in Hawaii? Is it like 2?
Something like that. It's pretty
Yeah.
pretty small compared to other areas.
You say 2 meters?
2 feet.
2 feet.
Yeah.
That's not very much.
No.
So that, but that definitely contributes to the movement of water even down here. Ooh, coming up on, there's a fish in Argus View.
Yeah, an Argus View. What is that? Can you zoom Argus View?
Oh.
This is a different.
Oh, that's a different one.
Yeah, it's a very different fish.
We haven't seen that guy before.
Okay, he's shy, but we got a little bit of a look at him. And we'll leave it at that. Fish, a very scientific identification.
Got it down. I didn't get it to species, but we got it to kingdom, so that's good.
Animal.
Yeah. But it wasn't eel-like, which is exciting to me because I asked Bruce Monday that question last night about why the eel-like form of these deep sea fishes is so dominant and successful, I assume. Haven't heard back yet.
Yeah, he's taking a while. I mean, he's retired right now too. So
yeah.
He'll get back to you.
Oh, I know. Yeah. Curious.
All right, where'd everybody go?
Where did everybody go? What's happened here?
Where did all the sponges go?
It's probably raining down there, so they all went inside.
Oh, yeah, maybe.
Those Walterii sponges, it looks that, you know, if you can miss them, could drive right past them, they like appear just translucent and sometimes hard to pick out of the background. Those stalk-like kind of little brownish colored spikes sticking up out of the sediment, or the rock, I should say.
Just to let everybody know, that yellow sponge that we collected here last dive, it's in the lab. It's in some ethanol for DNA, and there's also so much of it that we dried the rest of it. And it's just like cotton candy. It's so lightweight, and you barely even see any tissue.
Better go that way.
Fiberglass cotton
I just mean,
candy.
I better go that way. Like, stretch out a bit.
There's kind of a pretty ledge here.
A little fish passing through.
Going
well, look at that attachment of that sponge at the bottom of the screen.
There's a wall.
It looks pretty.
Let's see this sonar pass here.
Can we go to the wall?
I think it'll be okay. Yeah, 40 meters out, I think we'll be okay. It's a really cool rock, yeah.
Yeah, it's beautiful.
And we got some life back.
Yeah, well, this is an ideal current aggregating place. A nice little drop off there.
Yeah, it's kind of nice right on this little edge here.
I love how they park right on the edge, and then right just underneath the edge is where you see really high abundance usually. It's because it's I think because the currents are eddying there. They're going over the top of the ledge and then kind of swirling around behind it, so they get multiple shots at getting the food.
That's cool.
Filtering the food.
So a viewer is asking, in what kind of areas would these type of sponges most commonly populate?
Well, that's what we're trying to figure out. That's an excellent question, and that's a question we're asking ourselves. So what we're trying to do, and I can't think of any other way to do this, is to take guesses and go to a place and see, and sometimes you guess right, and sometimes you guess
Just
wrong.
rounding out will be good.
And then you take a look at your two type of sites and try to figure out what the difference is. But the only thing we can think of right now is they require hard substrate, as you can see, very consolidated substrate, and they seem to like high current with a lot of food. And that's it. But how you predict those from a map, you can kind of get the hard substrate from a map, but it's hard to determine where you can find the really strong food, dense current.
This should be fine, I think.
It's interesting they've been struggling so hard with these sediment cores, just how shallow the kind of the substrate is on those sediment areas. It's only about an inch or
There's
two
a coral
deep.
over here with a loop in it.
Coral with a loop. Oh yeah.
Can we get a zoom on the loop coral, please?
The loop coral.
Oh, just a trick of the
Just
eyes.
the bamboo.
Oh, that's got whacked there. Oh yeah, it's just a trick. Okay, thanks.
So the curly one is a bamboo coral, and the one next to it, I think, was in the family Primnoideae. Got a red star or something over to the left corner, but I don't know what that was. So you
That's
can
beautiful.
see the faces of these sponges are all facing in different directions, so the waters must be swirling right around this little promontory.
Yeah,
beautiful
look at
Argus
that.
shot right
That's
now.
really
That's
cool.
great.
Oh, sorry, can you zoom in on this guy right there?
Yep, you bet.
Is this the one I'm
Do
looking
you want to
for?
poke around this rock a while? Should I put a hold on the ship?
Maybe, I just need
Yeah.
a closer look at this.
Bridge nav, hold position.
Okay, good there. Go in, please.
There's also something, shrimp underneath it.
Oh, a red shrimp.
Oh, that's a sea star. A little sea star.
Can we go in a little bit further, or is
Yeah,
that
go
all you
ahead.
got? Can we?
I don't see you yelling Eureka. So.
No, I'm very close to requesting a collection here. Again,
back
because
the
we're in the
ship up?
monument, we don't do frivolous
Do you want
collections
me to back
here.
the ship up? I think Argus is going to swing over you, or maybe
we'll
you
be
can swing it
okay.
around.
I think we'll be okay, yeah.
Okay.
Okay, come wide, please.
Yeah,
Yeah, we can be on the far
think,
side of the rock.
I
It's not
think
so
this
tall.
would make a good collection.
Okay. Let me just confirm I can actually. Yeah, that rock's not so bad. I think we'll be okay.
You wanted to do some rock climbing, didn't you?
Mm-hmm. Not with Argus though.
Oh, okay.
Yeah, you can spin around.
So what do you think this is, Chris?
I can't hear you, Andy. It's either a polyopogon at a weird angle, which I don't think so because of these holes in it. I'm not sure. It could be in the family yymmdd, but it's bigger and a little different. Some of the members we've seen of that, I don't know.
Those are.
This is why we're going to try to grab a piece, because it's stumping us a little bit.
Oh, it kind of looks like that one, the hyaluronema.
Yeah. Except that osculum is really large. It still could be one of those.
Osculander's the hole on the top.
Yeah, or that opening.
Am I going to be able to make it in there without knocking a bunch of stuff? Let's see.
You know, pilot, I'm wondering, Andy's got some pictures up here, and I wonder if this would be just a really big version of something we've already seen. It's really hard to tell from this angle. Maybe we ought to do a pass on this.
Well, we can get one more zoom while we're here.
Okay.
And you can make that decision. Oops, no. Wrong button.
So for your armchair taxonomists at home, you could join us in identifying things by downloading the Okeanos Animal Guide, or not downloading it, you can view it online. Just type that into Google, Okeanos, Okeanos Animal Guide. That's kind of a version of that's what we're looking at here to identify some of these
zoom in, please.
Amazing animals.
So you want to pass or you want to keep it?
I think we ought to pass. I think we might be just looking at a big version of something we've already collected. Yeah.
Okay, well.
Yeah, thank you.
Roger, come wide, please.
Love
let me
that
take
shot
off
right
first.
there. It's beautiful.
Great.
I'm going to
Also looking for
again, it's gonna
corals
be
that we have, and I
130.
saw some urchins, by the way.
Oh, you have?
Yeah, on the previous watch, I saw some urchins.
Loop around this rock.
That would be very good. I saw a monoplacophorus, I think, which is a type of mollusk.
I think you can get a ship move going now. I'll be there by the time we're moving.
I
I'm going to
think
bring
they
it
said
back
they
to
saw an anglefish.
20 meters up, so I think, because it feels like we might do some more poking.
Sure.
Bridge nav. Can we get a 20 meter step, 130? Thank you.
Yeah, we would like to do some poking here.
Little sea star hanging out on the wall there. Whoa, that's gorgeous. Black coral.
This is really
Bathy
beautiful.
pathies.
No, that's overgrown. Those are zoanthids all over a primnoid or a...
The one down below?
The black one down below, you're right.
Yeah.
These yellow ones up on top are overgrown.
Wow, that's really cool.
Nice looking rock.
Yeah, it's beautiful.
Which of those again? The one on the top, the goldish colored one?
These
Not
big fans?
The big fans. Those
Well, there's two of them. The one on the right, I think, is a bamboo, and the one on the left is a primnoid, probably Paracalyptrophora. They tend to form these very, very big fan-like primnoid colonies like that. It's not absolutely certain. There's some other genera, which I don't know very well,
Quite the drop off.
that group of corals.
It's quite the drop off there.
Yeah.
Yeah, we're running right along the edge here.
Is that a Phariidae sponge? That one kind of in the lower center, middle?
Hard to tell from this distance.
Come down on Delta, please.
Bridge and nav, another 20 meters, 130.
Whee!
Ooh.
That upright one is a Tritopleura, one of those leaf sponges.
Oh, okay. Yeah.
Look at that up. No, that's just two of them. Okay.
I know that looked like a very unique form for a second, didn't it? Yeah, so we don't really know how old these sponges are because it's so hard to date sponges. You don't have things like rings, like coral growth rings, or easy ways to identify some of the components of the coral, even or date those. So just don't know, right?
Yeah, that's right. But I'll bet it's pretty old for some of these massive ones just based on their size.
What do you think pretty old
Could be
might be?
hundreds, if not over 1,000
Making you
years
work for
old.
it there.
Wow.
Making you work for it there. Up, down, around.
This kind of nice path you've chosen. There's a lot of stuff here on these rocks.
Oh, what was that down? Oh,
It's a big fan looking in the background there.
Yeah.
Oh wow, so Christmas tree size fan, it's huge.
Yeah, that's insane. Look at how tiny those lasers are. It's a large coral bush.
We could take a look at this a little close up just to make sure we know at least what family it's in. I think
Hard to
this
get a
is
scale of that, really.
Yeah, it's a monster. I think this is a bamboo coral.
Let's get a zoom in and check out some polyps or whatever.
Yeah, exactly. There is a commensal up on the left corner of it too. Yep, it's bamboo. And I'll look and see where the branch points are because that's usually informative.
hard to tell. It's so,
Yeah.
So dense.
tangled up like
Yeah.
that.
You can see the nodes and occasional view in amongst the branches, the nodes of the black bands around the stalk. I think it's an internodal that is branching not in the black bands, but between the black bands.
Oh, was that a fish down in lower center? Or is that squat? Oh, just a basic brittle star.
All right, come wide, please. I'm going to do a
Basket star.
look at the whole thing action here.
Yeah.
Oh, look at us way far away.
Yeah, I wonder if we could just come down a little bit further and get a closer look.
Are you looking for more close-ups, Chris?
What's that
Yeah,
thing?
if we could. I gotta look at my
that's a
guide
pretty-
and see. I think this is something.
Zoom in,
we've
please. We'll
seen
check out that thing
before.
that was just
Crinoid?
What's
Yeah,
that?
it looks like it.
Yeah.
I love that crinoid.
Nice
Ooh,
colors.
very
So
purple
cool.
vase.
So this is a feather star. And if you can see how he's wrapped his cirri, or that's the equivalent of their feet right around that branch. And you also notice that the tips of the arms are bare.
Yeah.
And those are some of the diagnostic features when you look at. This is Xenometrid, and it's Cyathometra is the name of the genus. And because it's gone all the way to genus, I'm pretty sure we've collected it before, because I don't think we would know that if we hadn't collected it.
Well, it's definitely demonstrating the name of the game down here, to get up high into the water column so you can access more current and more food.
Yeah.
It is all the way up at the top of this very large colony.
I try to look at this coral, see if I can. Okay. Okay, this does look like what's called an ecnomysis. If we can find a spot where the polyps are closed up, if I remember correctly.
I don't know if we'll get that lucky.
Yeah.
Come down in delta, please.
This particular
You can go,
genus
yeah, go for it.
has what's called volcano
We'll
polyps.
keep coming down in delta.
Volcano polyps.
Volcano polyps.
Come wide on Herc, please.
Ship is stopped now, but it is still pretty
Roger.
far off you.
I gotta go back to this side.
Well, I think I'm going to try to get a really good picture of that crinoid, because the ones in the guide often don't include the naked tips. And if that's a diagnostic feature, it'd be nice to have a better image of that. Is this the one you're thinking, Chris?
Excuse me. Yeah, you can see the naked tips right here.
Oh, okay, yeah.
So that's the guy.
Zonometridae.
I called it Cyathro. It's Cyathrometra. I said Cyathometra.
Cyathrometra. It's beautiful.
Yeah.
Yeah, we haven't been seeing many crinoids on this cruise in general.
We've seen some. These feather stars often will be found perching on rocks. They don't have stalks.
Are you going to want a sample of this at all, or should we keep moving on?
I just wanted to take one last final shot at seeing if I could see closed-up polyps. Maybe this branch point right here, oops, right up there, might yield some closed-up polyps.
Okay, go ahead and zoom quick, please. Oh, I lost it. Nope, I guess we're going.
Okay, that's fine. I'm pretty sure it's Ecnomyces. It is a bamboo coral.
Yeah, you can come up now.
It's almost a dead ringer for the example in the guide.
I'm back wide on Argus.
That's for Kainalu too to take a look at.
That's all good. I stuck around far too long. Pretty what? Yeah. I get scared with the tether being on top of Argus.
That's
I've heard rumors of it being scooped off of Argus with the manip before. I do not want to repeat that. Oh, I gotta see something here. Stay where you are a sec. I want to turn off some lights here.
That's cool.
Yeah, yours are good. This is what's lighting up this right now.
That looks so cool.
I gotta get that solar lens flare out of there.
What is lighting this up? I don't.
Say again?
What is producing the light? Is it
That's
from Argus?
Yeah.
Okay. It's rather eerie looking, isn't it?
It so is.
You can see the shadow of the tether bouncing against across the bottom there. Over the bottom, but the shadow.
Oh, that's pretty cool.
That is.
It's a little bit closer to what it looks like, except it's darker, but in real life, it's completely dark.
But it definitely gives you better kind of feel for the environment. Dark, cold, and a lot of pressure.
Yeah. All right, I'm gonna turn the lights on and go back to normal.
That was kind of cool, thank you.
Do you want to wait till you're back out in front?
I think we can go now.
Okay.
I'll get going.
Bridge nav, 20 meter move, 130.
Coming up to a nice little rock here.
Well, our viewers are certainly having a great time.
Oh, it's
looking
terrific.
at all of this diversity of sponges and corals.
You know, I'm having a pretty good time too.
Yeah, one of our viewers of 4 years says it's still amazing to watch this. After 4 years, muddy. Frank from the UK also is thanking us for all of this.
I agree, Muddy.
He's also asking, when you come across a large field of sponges as today, do they have a collective name?
Well, the majority of the sponges we're seeing are probably, well, they're definitely in the same family, not all of them, but most of them, and that family is called yymmdd. And then many of the really massive, huge ones are in the genus Poliopogon for now. They don't have common names, I'm afraid. The only common...
Was the question about the the collective noun, as in like a group of sponges or a school or something like that?
Or a garden of sponges?
Oh, I see. A herd? No.
Since they're animals, right?
Yeah, that's right. But no,
a murder
herd
of sponges.
herd wouldn't work.
I love
What would you
collective
call
nouns.
a group of sponges? That's a good...
What's that?
And you do say coral gardens, and I think they use the term sponge garden, so I guess a sponge garden would be appropriate.
I think they need their own little quip.
Well, we think of kitchen sponges, so a sponge
How
bowl,
about a sponge soiree?
A sink
nice
of
rock.
sponges?
a sink of sponges. Look at this rock.
A soiree of sponges.
Nice spongy rock up ahead.
Yeah.
Mm.
Ask our viewers what they think we should call a collection of sponges.
Sponge bed? Is that what Aaron in navigation said?
What? No. I did not venture a guess. I'm just admiring the spongy rock.
I'd be curious to hear what it is.
I don't think there is a term for it.
You don't think so?
No, I mean, because up in Alaska where they have these immense sponge beds, I mean, you could call it a bed, I suppose, but
There's gotta be one.
we could take a look at that yellow coral if you like to.
Yeah.
This is actually a bamboo that's overgrown, and I just don't know how many times
Sure.
people have imaged that. It's a cnidarian on a cnidarian, and it's killing its relative. A little chrysogorgid there, front and center, too.
Okay, zoom in there, please. Start with this guy and move over.
Beautiful precious coral, Coralliidae, right here, the red one. I think this is overgrown. There's yellow varieties of bamboos, but I think this one's being overgrown. Yeah, it is. So the type of, it's not, I wouldn't consider it a coral, but the type of cnidarian that is overgrowing it is called a zoanthid or a zoantharian. And so most of the original animal, if not all of it, is dead already. Maybe a couple live polyps down in amongst those branches. So the zoantharians, almost all of them cannot secrete their own skeletons. So they've solved the problem by stealing the skeletons from other corals, the overgrowing them.
Imagine being a skeleton thief.
That's what they are.
Oh, there's a nice shrimp, red shrimp on the side.
That shrimp was one of two types, but I don't need to throw out another tongue-twisting scientific name.
Well, thank you to all of our viewers sending suggestions for a group of sponges. And it would be really nice, too, if you write your name.
Hey, science. Sorry to interrupt. Anything you want to look at more here? Should we just carry on?
Yeah, let's go ahead and carry on.
Okay.
Thank you.
Reg nav, 20 meter step, 130.
So these are some suggestions for the group of sponges. A squish of sponges,
They're not all squishy, though.
sponge bed, sponge garden,
Uh-huh.
a sink of sponges,
Yeah.
a filter of sponges,
A filter. It's not bad. I'm not sure it actually works, but it's interesting.
a filter, a field of sponges.
Field of sponges,
Mm-hmm.
maybe?
So I've heard suggestions, but I haven't heard what it actually is. Is there really not a
No.
collective noun?
I don't think
There
so.
isn't.
I
I
don't
was
think
trying
there
to
is.
search for one, and there isn't. An
That's
answer.
bizarre.
Yeah.
Yeah.
Are
Ah, the poor dejected sponge. It doesn't get its own group.
I guess for shallow water stuff, you could have a reef of corals. Could you get a sponge reef?
Yes, you can.
So
The
that wouldn't be with
Caribbean.
these deep sea ones, right?
It would. It's again more applying to some of the things you see up in Alaska, where the entire substrate is. I mean, you don't even see rocks in between. There can be sponges just right up against each other. Where
You get that off the coast of British Columbia too.
yeah, British Columbia as well. So we're not seeing that. They're not totally covering substrate. They're just a lot of them sprinkled around.
Or even the shallow reefs like in Fiji or like the Caribbean, you can have areas that are heavily dominated by sponges.
Now Scott was interested in these leggy bamboos. You got one right at the bottom there.
I could take a look at that one.
He
Yeah.
wanted to look at the nodes, I think. I don't know if this is the one that he's interested in.
Can give it a gander.
This is Scott France from the University of Louisiana Lafayette, who's very interested in these bamboo corals.
Okay, zoom in on the coral, please. See if I can get that branch area.
Okay, this is what's referred to as volcano polyps, because they kind of, when they close up, they form these little mounds that look like volcanoes. This one looks like it almost is branching from a node. It's really hard to see. I just don't know, and he's not on the chat right now. For some bizarre reason, he's taken a Thanksgiving weekend off.
Weird.
Yeah, weird guy, jeez.
So what causes them to close up?
Don't know.
ROVs.
What kind of coral is this?
This is a bamboo coral. It's a sparsely branching bamboo, and it's really hard to see whether it's branching at the nodes or branching at the inner nodes. But thank you for that.
And we have one of those polyepigon sponges that made a poor life choice.
Bring it to 120.
Sure.
Left of middle there,
Bridge
swapped
20
over.
meter step 120.
Yeah, I see what you're talking about, and you're right. Even though you're saying it humorously, that's exactly right. The rock was too small that it landed on, so kind
Bridge, can you make that 120, please?
There's a moral in there somewhere. If you're going to attach yourself to a rock, make sure it's a
Great,
big one.
Yeah. Where's our newbie? Where is our brand new species here? Come on, I know you're here. I saw you the other day. I saw a couple of you.
So in these Walterii sponges, like the one in the lower center, what purpose would those fragile tendrils coming off the central core serve?
Well, what do filter feeding organisms try to do? To
create their own current.
Yeah, but also what do they do? It's sort of like corals too. Why are they big branching fans? It's because they're trying to expose as much of themselves to currents, because they have a better chance of catching stuff, more food. So a lot of those lateralia, which is what they're called, perhaps that's what they do. Okay, that's just a smaller pot-bellied sponge down there. This is a really nice track because we know this is not where we were. We were a little further down when we were the first dive, so we're actually looking at different parts of this ridge.
Yeah, just off the side of the ridge.
Yeah, it's a good track. It's very useful.
One of our viewers wants to know, why are some corals plain white while others have color?
I imagine, excuse me, I imagine it's the it's usually tissue that's colored, not the skeleton, although there are exceptions. The coralliids, that pink one down below, the actual hard parts can be colored, but it's not really sure. It's just that
Bridge
we're
nav,
providing
20 meters,
artificial
130.
light, and the tissue in their bodies and different species can reflect light a little bit differently, giving them different colors, I guess, my guess.
Very different than shallow water corals, which get their color from the commensal zooxanthellae that live in their tissues, right?
Right.
We don't have those down here because there's no light.
Right.
So what I'm seeing here is that the corals and sponges are thriving pretty well, even though they're not right on the precipitous edge of the ridge. They're just on the slope, on the steep slope.
Yeah, but they're very close to the edge, and we've done these. We did a lot of them in the capstone project, and if you drop like 50 meters or even 100 meters down, they all peter out. They're still there, but not anywhere near the numbers.
Right.
This is kind of nice just to run right along this edge.
How far are we from the ridge we were on last dive? 100 meters or so?
Let's see. 120 meters. Yeah, 100 to 120 meters.
Okay, so not super close. It's a good wide field anyway.
Yeah.
Luca from Germany is asking, how do these sponge fields form? Why do they appear to show up in groups or lines even?
I don't know. Again, I can only speculate on that. And it's possible that, I don't know much about sponge reproduction, I'm afraid, but it's possible when they reproduce, they release competent larvae. That means larvae that are ready to settle. And there's some species of fish and corals and things that don't do that. They actually release the sperm and the eggs that fertilize in the water column, and then it takes a while for them to form larvae, and those could drift off away from a site further. But ones that actually release their larvae that are ready to settle, they could just drop down and settle right next to their parents 1 meter away. And that's how you could potentially, over time, build up a pretty dense community. That's a guess. I don't know how else. The other possibility is that the larvae themselves are sensitive to chemicals. It's called chemotaxis. And so they would tend to settle out where they sense there are other members of their species around. So those are two things. Third thing could be simply a physical environmental thing where the way the water flows over this, it tends to aggregate things, sort of like trash piles, if you will, out in the water, where it aggregates things.
Like the garbage patch.
Yeah. So
Sponge patch.
it's a good question.
So
This one looks very ominous.
It does.
Looks like a hoodie or something.
It does.
Oh, what was the next
Or
one?
gnome hat.
Ewok.
Yeah, one of our regular viewers, Jason, says that he personally likes coral forest and sponge gardens as the group.
Coral
Okay.
forest is actually, it's catchy, yeah.
Yeah, I get where he's coming from. Interesting.
How about a bob of sponges?
A what? A bag of sponges?
Bob.
A bob of sponges.
Oh, a bob of sponges.
Group of sponges. Bob of sponges.
Yeah, by the way, that sort of yellow one on the bottom, that's a dead sponge, or one that's dying for sure. It's not just a different one. The yellow one that we collected the other day wasn't brown like this. It was yellow, and that was the color of the tissue, I think. Then I'll get that home, and I'll find out it's just one of these massive ones that was dead, and then I'll be crushed, like you're saying. But I don't think so. Really looked at those pictures, and I think we have an unusual.
Yeah, it
unusual specimen.
It's very unique. So we are coming to you live from Papahanaumokuakea Marine National Monument and UNESCO World Heritage Site. And we are on Don Quixote Seamount. Oh, we're going to look at
What do you want over there?
Sponge.
Yeah, the guy that's flat against the rock, if you could, please.
Is it
As a imaging or sampling?
Imaging first.
Roger.
bridge nav hold position?
It's just the angle, I think.
Yeah.
I'm sorry, it is just, yeah, go ahead and zoom in. But
We'll zoom and move the ship, yeah.
Yeah, it is, I'm sorry. I thought it was flat against the rock, and it isn't, just the angle.
Okay, bridge,
Roger.
rather 20120, thank you. No worries, rather safe than sorry.
Yeah. I am desperate to find something that's novel that would be worth a collection. Again, we are in a monument, and we do not do collecting lightly.
Do you remember where it was? We could just go back there.
I don't remember.
Hustle.
In fact, I tried to find the picture, and there's a lot of them in that folder, and I didn't see them. I didn't get through the folder, but.
Roger.
Well, it was very close to the yellow sponge, the new yellow sponge.
Yeah, I think there. You remember the one I'm talking about?
Yeah.
Yeah.
Well, kind of.
There's a fish, I think, going up the wall.
Up the wall. Where is it?
He went off.
Off the wall.
He's on the right side, there's fish.
Right side.
To your starboard.
Chase him down, Trevor.
Yeah.
Go get him.
I don't have much leash that way, but I was hoping it was left. Where are you?
He went out the bottom.
Ah, there it is. I'm coming for you.
That's another one.
Different fish.
Yeah, but that's
well,
okay.
you know what? This is a fish.
That's okay.
This is a fish.
It still qualifies.
Oh, that's a
Hey.
silver fish.
Creepy fish.
Can we do a zoom? Sometimes they swim off really quickly.
Yeah, stand by. Okay, go ahead.
Oh, very nice.
Keep working my way closer as well.
Ooh, what kind of fish is that?
This is a cusk eel.
Good looking fish. See ya.
There he goes. Well, it's probably in the family Ophidiidae.
Where'd you go?
That's it. That's all further I'm going to go. Not sure about that one. Oh, Asako Matsumoto, sorry, I'm neglecting the chat. She says that you can actually find soft corals in Guam with zooxanthellae next to the beach. So, and those are octocorals that have the commensal algae in them. So that's very interesting. I didn't know that. Thank you, Asako.
All right, I gotta not go this way anymore.
Okay.
I gotta go the other way. Hope I'm making some people seasick by looking at that quick turn. Where's he going? Ooh. All right, there we go.
If we want to go up that steep bit, we can bring it to 100.
I mean, yeah, it's up to science. I'm happy to keep side hilling. Going up the steep bit probably brings us closer to our old track.
It does, for sure. But we're getting to the point where that's what we'll do.
Or
Or we stay along this area.
Yeah, Roger.
Yeah, right where Aaron was sort of pointing right there. Just right below our old track would be good.
So can you see, I know it's hard to see on high pack, but something like that?
I can see that. Yeah, I think that's okay. That would be fine. Thank you.
Great.
All right, that's going to be 105.
Roger. 105.
Bridge nav, can we change that 20 meter step 105?
While we're here, could we look at the coral that's having a bad hair day?
You want me to look at the bad hair day, Roger?
Right there,
The
thank
heavy
you.
metal coral.
Okay, zoom in, please. That's a great hair day.
So even
Don't listen to them, Coral. I think you look wonderful.
Well, nothing's eaten it, and it's all intact. It's really good shape. So this is different than that other bushy one that we saw. This is a pinkier one, and I think it's got different polyps on it. But I don't know. I can't quite see where it's branching.
I can go back to it.
There's a primnoid. That's Calyptrophora angularis.
No,
I could already tell.
Do not kill anything.
Did we ever get a determination on what some of that golden kind of dusky stuff is on the bottom that we've seen in other areas where there's sand sediment?
Yeah, we determined that it was golden dusky stuff.
Oh, okay.
We brought some up, didn't we?
Good. Did we?
What's that?
I thought I saw some in one of the samples.
Oh, I guess we did. It was on one of the rocks. That's right. So
Yeah.
Huh. Is it some bacterial thing or unknown? Unknown.
Unknown.
I guess that would be a Beth question.
Yeah, I did ask her. She didn't. Maybe I got to show her a better photo.
How heavy can these sponges get?
Well, they retain a lot of water. So let's just say if Trevor was able to actually grab one of these massive sponges and bring it up, it probably wouldn't be so bad in water, but as soon as you got out of water, the thing would weigh a ton. So, and of course he can't, but a lot of them just, when you grab these massive sponges, they just are, actually have to squeeze them out just like you would a kitchen sponge if you're going to put them in alcohol. Otherwise, you're going to burn a 55-gallon drum of alcohol for one sponge. So I don't know if that answered your question, but it's and then when they totally dry out, they're very lightweight. They're not that heavy. Kind of, I think, a dead sponge base on the face of that rock.
Oh, I love that.
Should mention something about the rocks. Is some kind of a pillowy formation.
Uh-huh.
I think it, hard pressed to believe it kind of erupted right from there, so it might have been a chunk that fell down slope. It was actually, the eruption was actually further up slope.
Seems like there's a lot of these kind of rocks, but this just happens to be the biggest one, which makes it the best spongy one.
Yeah.
Oh, there's some of those waves. Some of the patterns from the.
Oh yeah.
From the waves.
Cool pattern from it too, isn't it? Like.
Yeah, zoom in on those if you can
make that happen. Yeah.
Because we were really interested in those. And the reason why we were really interested, we had a really great discussion yesterday about there are waves that are, or energies dispersed through tidal patterns, but there are also what are called internal waves.
Please. You want to look really close at them?
Sure. Even though I have no clue how to interpret this, but.
Yeah, or probably the wider view that shows the. So essentially what's interesting about this is that those patterns, like you might see in a shallow water on a beach, just in the shallows where you can see where the waves have caused a distribution pattern in the sand, the same thing might be going on here with these internal waves, which are deeper in the water column, that are causing those patterns in the sand. So it just is potentially indicative of some kind of deeper flow that may contribute to why these communities are so dense here, because there's more water movement of dense water going up and down, and therefore moving more material past these filter feeders to scoop out of the water, filter out of the water. So anyway, just kind of neat. We're still trying to understand that and seeing if it's actually what's going on in these communities, these seamounts.
I don't know if anybody in the science row has seen, but John Hughes Clark is recently been doing a lot of stuff with internal waves. You could see it in multi-beam in the water column. Super, super cool.
Oh, wow. That's cool.
But you have to have John's amazing skills for digging into the data with his own code. It doesn't show up. And you need a pretty high res sonar too.
That's because of you can see the change in density of the water, I assume?
Yeah, because it's a density difference. So
yeah.
he's imaged like those, are they Kelvin-Helmholtz waves? Or I'm not sure if I'm saying it right, but he has beautiful, perfect images of that in water column data.
That's cool.
Well, what does it look like? It's in the water column display in the multi-beam?
Yeah, he can pull it out of the water column display, but he's doing it with really pretty high frequencies, and just he has his own suite of tools to really tweak it out, but it's amazing.
Wow.
Yeah, so if you're curious about that, just Google internal waves, and you can try to understand what we're talking about. But like for the Hawaiian Ridge, what you can see is these deeper water currents moving water, bouncing off the Hawaiian Ridge and radiating back out on both sides of it because of the water impacting the ridge and then radiating back out again.
Pilot, any chance we could get a quick look at this guy down there?
Yeah, no problem.
Thank you. Again, I'm trying to find this coral that Dr. Scott France thought he saw here. I'm kind of keeping an eye out for some of the things he was interested in.
All right, zoom in, please.
Get, yeah, a little bit tighter.
Is this all you've ever wanted?
Yeah, this is fine. I don't think it's the one that he thought he saw, so thank you very much.
All right, we'll enjoy the look and then move on.
Sure.
So that's another type of bamboo coral.
Yeah, it's the one he thought he saw it's called Acanella weberi, and it's a nodal brancher, if I'm not mistaken. And the branches tend to come out very low near the base and almost a whirl.
Or zoanthids in the back there.
Yeah, zoanthids on a paragorgia. There's a leaf sponge there on the left.
Good looking leaf sponge, yeah.
Yeah, those are really neat. I mean, just amazing all the different forms that these organisms take to essentially collect the same. They're all going for the same resource, but they've adopted different strategies to collect that resource. And it's really kind of fascinating how they adapted to different filter feeding mechanisms.
Yeah, there's a lot of different ways to do that apparently and do it successfully.
Yeah. And the sponges create their own, once they get the material inside there through their pores, inside they have the cells, certain cells have little flagellae, little tails that whip and actually drive currents inside the sponge. And there's a really cool YouTube video about somebody who's studying this and using dyes to study this, and they inject dyes around the sponge, and you can see how it moves through the internal structure of the sponge, the green dye. So definitely check that out. It's really interesting to see, because it's not just a straight flow through. It gets in there and creates all these little vortices and other things that keep the water inside and move it around. They get more opportunities for filtering out organisms out of the water, and then it comes out the other side of the sponge. So definitely take a look at that. It's beautiful.
Oh, what is that? A hairy worm?
No, it wasn't a worm. It might have been a little salp or something like that. Hard to say.
Where are we looking?
Oh, is this something drifting by in the water column?
Squishy.
Squishy.
Squishy. The video is called Sponge Dye. By C Studios is one of them. It's really cool.
What's that little pom-pom on a stalk? Is that just a teeny sponge?
Oh, I don't know, but it could be anything. Can we zoom in on the pom-pom, please?
It's a lollipop sponge.
Yeah. Cool. Oh, it's just a little wee guy.
It's a very young.
No,
what
it's
are those
not.
again?
No, it's not.
What is it? Don't hold
Oh,
out on us,
sorry, come
Chris.
wide a sec.
Yeah.
I'm
Well,
just-
I mean,
try that
it's
again.
not a collectible, but we've been talking about how the stalks insert on different families of sponge. Now, Andy, I want to see how well you did take your lesson. Have you been studying at all?
I'm going for Euplectidae.
Oh,
Stalked.
Andy. Oh. Afraid I can't give you a passing grade.
Oh, come on, but it looks just like a
Okay, zoom in,
bolosoma.
please.
This
I'm going
sponge
to try a camera.
is in the
Yes.
genus Calophacus, and it is not a euplectellid.
Oh.
It is a rosellid, a stalked
It's a stalked
rosellid.
Rosellidae.
Yes,
Oh, I
If I
If I
see.
bump up, we
I
can get
see
one
my
more
error.
zoom in. Go ahead, all the way. Yeah.
Calophacus.
that's beautiful.
Oh.
So is it really young?
No,
Oh,
it's just that this is a variety that it's just not like those big massive ones. It's a different species.
too bad.
Yeah,
I want to see a baby one of those. I think that would be super cute.
I'll bet we could. Maybe we'll encounter one
Oh,
before
a
the end of the
baby.
cruise. I'll bet.
All right, come
You
on,
gonna
please.
adopt one? Adopt a baby Calophacus.
We'll just have an encounter with one.
That's a nice variety of sponges there, though. You got the Walterii, the Calophacus, Bolosoma, all in one spot. Little squat lobster parked on top of that
Oh, yeah.
Walterii,
Good eye.
just right of center there.
So a couple of our viewers want to know how large
no,
in
it's a-
size those hooded sponges are.
Well, there was one back in 2003 we came across that we called, before we knew its name or even genus, we called it Jabba the Hutt.
And how big was that?
He was probably about
Big.
2 meters across by about 1 to 1.5 meters tall and about at least 1 meter deep or so.
That was the one the size of the minivan found on the
No,
capstone cruise?
that was really big. I mean,
Yeah.
but that's a different species we haven't seen. I think the viewer's asking about these sponges,
Oh,
how
okay.
big these kind get.
Well, you can measure them yourself when we got the lasers on. The lasers are 10 centimeters apart between the little green dots there. So like this one, we're about to hit the base of that. It's like, what, 15? And across the top is about 15 centimeters.
And that's the slender view,
Slender view, yes.
side view.
Across the front, it's probably like 30 centimeters for these small ones.
So a little comment here. Some people might be wondering, well, what's the point of just sort of cruising over the bottom if we're not collecting specimens? And I can tell you what the point is, because all of this video, when it gets back after the cruise to a laboratory in Hawaii, there's a team over there. It's called the Dark Lab, and they're going to annotate this video, which means they're going to extract every drop of data that they can get, and that means counting all of these sponges as best they can, even measuring them, the whole works, and creating an immense thousands and thousands of record long, thousands of records, a database that then is going to be submitted to the Deep Sea Coral Research and Technology database, and that will be posted online in its ever-growing enormous database on corals and sponges. And so we are collecting data right now, we're just not collecting specimens. So we're not just sort of cruising and touring the bottom here. It's just another product that we get from these dives.
Well, these sponges are lucky. They're in a fully protected area in Papahanaumokuakea Marine National Monument, And NOAA is actually considering this area as designation for a national marine sanctuary, which will provide additional protections for that place. And we have some public meetings coming